Welcome, everyone. Thank you for joining us for today's keynote presentation, Nucleic Acid Detection with CRISPR Diagnostics, presented by Dr. Omar Abudayyeh, who is an MIT McGovern Institute Fellow, and Dr. Jonathan Gutenberg, who is also an MIT McGovern Institute Fellow. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. And if you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Abudayyeh and Dr. Gutenberg. I will now turn the presentation over to them.
Great. Thank you for the introduction. So Jonathan and I will be talking about a lot of our work that we did as graduate students in Dr. Feng Zhang's lab at the Broad Institute. I will sort of speak for the first half about sort of the early CRISPR discovery work and development of the SHERLOCK technology for diagnostics, and then Jonathan will close in the second half with more of our sort of next generation feature development and where we're going with the platform. So to dive in, I just want to have a conflict of interest disclosure. So Jonathan and I are both co-founders of SHERLOCK Biosciences, which is commercializing the SHERLOCK platform, as well as advisors for Beam Therapeutics. So a lot of the work that we do is really motivated by looking towards nature for tool development. And nature has been really remarkable, adapting unique systems and proteins for all sorts of sort of environmental challenges that bacteria and other organisms are exposed to. And just to see how rich of a resource nature can be, we can look at many examples where normal sort of systems in these organisms can be adapted for really powerful applications, including sort of the original innate immune systems that were found in bacteria restriction enzymes, which are really useful for microbiology and cloning, fluorescent proteins, enzymes that are useful for energy or even cleaning like cellulase, and even viral vectors for gene therapy and optogenetics for modulating the nervous system. And of course, what we're here to talk about today, the CRISPR system, which is an incredible adaptive immune system in bacteria, has been sort of ushering a revolution for genome editing. And so, one of the questions for us is, how can we discover new CRISPR tools and things that might be useful for totally new applications, for example, diagnostics? And so when Jonathan and I first started in the lab, everyone was really excited about Cas9 and how Cas9 could be used for programmable DNA cleavage. And we had this really interesting question of whether Cas9 was the only thing that was there. So, of course, the CRISPR system is very diverse, as you can see here. All these different, proteins and colors represent the diversity of enzymes in the CRISPR universe. And of course, Cas9, if you zoom in at the bottom, is really just a small slice of this universe. And so we had a question of whether we could find more enzymes like Cas9 that are simple, just a single protein, versus these other systems which have many proteins and that can be adaptable for different technology purposes. So to do that, we can simplify all that diversity on the last slide into a really sort of condensed set of features. And here you can see the different classes, sort of expression, interference, adaptation proteins are kind of colored. And what's really interesting is if you look at the adaptation proteins, which are shown in blue, they actually tend to be the most conserved enzymes out of all CRISPR systems. And so we can actually take advantage of that conservation to pull out new systems. And so we thought, could we take Cas1 and use that as a seed for a computational program that could look in every bacterial genome that's been sequenced for new systems? And so the idea here is we can take Cas1 and search bacterial genomes for every single sequence or area that contains Cas1. And then we can look around the Cas1 in the genomes for other proteins that might be near it. and start to sort of cluster those proteins and look for proteins that tend to be enriched near Cas1. And using that enrichment, we can start to hypothesize what a CRISPR system would look like around Cas1, and then we can build new CRISPR families from that. And so we can pull out the candidates from this pipeline, and we can sort of analyze what they look like. And so when we did this analysis, we actually found many new candidate systems that were sort of centered around Cas1. And so one of the ones was this type 5a prefran, which is now called CPF1 or Cas12. And it was really exciting because it was the first single sort of protein beyond Cas9 that was shown to be useful for programmable DNA cutting and genome editing. And then these type 5b and type 5cs are Cas12b and Cas12c, which are also useful for DNA genome editing. And then what was perhaps most exciting was this last one, this type 6 locus, because this sort of pink-purple protein C2C2, which is now known as Cas13, actually looked like it cut RNA instead of DNA. And the reason we suspected that is because the C2C2 enzyme has these domains shown in dark purple called Heben domains, which are typically RNase domains instead of DNase domains. And so we did a lot of work, both in bacteria and biochemically, to actually publish a paper showing that C2C2, or now it's called Cas13a, it was actually a programmable RNA cutting enzyme, or RNase. And the way it works is shown in the slide, where essentially there's a single Cas13 protein that can complex with a guide RNA. And this guide RNA is essentially primed for cleaving invading RNAs like a phage RNA. And it can cleave the RNA genomes of these invading sort of viruses. And the idea here is, instead of cleaving a single cut like Cas9, it will generate multiple cuts in single-stranded regions of an RNA, and it can actually eventually degrade RNAs. So it's a very powerful enzyme that does a lot more than just a single cut, which is what made Cas13 really unique. And where that becomes especially powerful for diagnostic applications is this additional feature of the biochemistry, which is this idea of promiscuous cleavage or collateral cleavage. And what we found was that when we incubated Cas13, but without a target, so that the guide RNA had no target, you would obviously add Cas13 and see no cleavage of any RNAs in solution, even if this green RNA is not targeted by the guide. So it would just stay intact. But what we found is when you added the target RNA such that the guide RNA would recognize it and activate Cas13, we saw not only cleavage of this black target RNA, but also cleavage of other RNAs in solution that were not complementary to the guide. So it looked like basically the presence of a target could activate Cas13, and then Cas13 would just chew up any RNAs in solution. And we realized that that activation activity is essentially reporting on the presence of a nucleic acid target. And in essence, we thought maybe we could turn that into a diagnostic. So the way we tried to adapt this for diagnostics was we realized if we added an RNA reporter with a fluorescent probe on one end and a quencher on the other, this RNA probe would be not fluorescent, but when cleaved, you would separate the fluorophore from the quencher and you would get fluorescence. And so the idea here is if there's a blue RNA in a sea of orange RNAs that you want to detect, you could design a guide RNA for Cas13 that detects it. And when that guide RNA binds to the target and activates Cas13, you get cleavage of these reporter RNAs, which release fluorescence. And that worked fairly well. We were able to get sort of, into the low picomolar, maybe high femtomolar range of detection. And so that's pretty neat. But it's still, in the order of millions to hundreds of millions of molecules per microliter. And so for a lot of clinical applications where you require higher sensitivity, you actually need lower detection limits. And so we developed a way to sort of improve upon this initial collateral detection. And so we came up with this technology, which we essentially had this backronym, SHERLOCK, that we called it. But the idea here is you can take in DNA or RNA, And you can isothermally amplify it using this technology called recombinase polymerase amplification. And RPA is, you can kind of think of it like PCR, but instead of needing to thermally cycle the steps, you can actually use enzymes to replace those steps. So there's a recombinase that can anneal the primers and a polymerase that can extend the primers. And so the whole amplification actually operates at 37 degrees, so at a single temperature, which makes this really convenient. Then through the amplification, we add T7 transcription handles so that we can transcribe the amplified DNA into RNA, and then this RNA can, of course, then be sensed by our collateral detection method. And what's really great about this Sherlock approach is all of these steps can operate in a single reaction. And so they're all compatible. They all operate at 37 degrees. So it's actually just a single step, really simple buffer that can detect nucleic acids. And so when we tried this out, what we found is, of course, Cas13 by itself, like I said, had low picomolar sensitivity. So this scale is an attomolar, and attomolar is 10 to the minus 18. And 2 attomolar, which is at the right end of this x-axis, is about single molecule sensitivity, single molecule per microliter sensitivity. So when we add this pre-amplification approach on the last slide, which is the Sherlock technology, you see that the whole curve comes up, and we actually can then get down to 2 attomolar detection. And this is on RNA, but we can also do this on DNA as well. So a really powerful method that gets you quick amplification. A lot of these assays are, about an hour. So that was a really great first demonstration that the technology works on synthetic DNA or RNA. And we got really excited about what applications in clinical diagnostics could we then go after with this platform. And so, molecular diagnostics are really an up-and-coming area. The molecular diagnostics tend to focus on proteins and sensing proteins using antibody-based assays. Nucleic acids even better because it's easier to design an assay. You just design a guide RNA or probes. And there are a lot of applications for nucleic acid diagnostics, including viral detection, being able to detect bacteria like in sepsis. A lot of efforts to make these types of assays really portable, which what we've been thinking about with Sherlock, as well as even genotyping, detecting cancer, and even beyond healthcare, there's a lot of agricultural applications as well. So on the next set of slides, I'm going to talk about a few of these areas that we've applied Sherlock. So the first area we kind of went into was Zika detection, because at the time, a couple of years ago, this was a very big problem, and it still is a problem now. And so what we did is we packaged Zika fragments into lentiviral particles so we could work with the viral particles. And we showed that with just a simple heating step, we could break open the particles and then put them into the Sherlock workflow and get detection in less than an hour. And so we were able to actually get down to 2 attomolar of viral particles, which is a single particle per test, and that we could detect Zika very specifically. So dengue has similar sequence to Zika. And so it shows that our test is very specific. We also did this for human samples. So we took human serum or urine from patients that were infected with Zika, and we could actually put them also through the Sherlock workflow. And we found that we could get close to single molecule Zika detection. So each bar here is a different patient, and they had different loads of Zika in their bodies. And so we have a sort of range of concentrations we were able to detect in these people. So that was really great. And then we also wanted to show that we could theoretically make this a portable test. So in our early versions of portable Sherlock, we actually could freeze dry the Cas13 detection reaction on pieces of paper and then reconstitute the sample on those pieces of paper. And we found that we could still get into the attomolar levels down to 20 attomolar or 10 Zika particles per test. So that was also really promising as well. Now, lastly, for these early applications, we also wanted to show that we could do very good specificity on nucleic acid. And so I mentioned a little bit earlier that we were able to distinguish Zika versus dengue, which those targets tend to differ by maybe 4 or 5 positions. But we wondered whether we could detect a single mutation. And so an interesting application for that is cancer mutations in cell-free DNA from the blood, so that you could just do liquid biopsies and then detect mutations coming from tumors. A common mutation is the V600E mutation in BRAF, which is present in melanoma. And so we designed a Sherlock assay that could detect this mutation, as shown. The sequence is shown here. And we, of course, had a CRISPR RNA that detects the wild-type BRAF, which is a normal signal, and also the mutant sensing, which is the tumor oncogenic version. And the samples we had varying levels of the V600E mutation. So as shown here on this graph, there's 0%, 0.1%, 1%, and 5% loads. And so, in the blood, obviously, if you have a lot of tumor, you'll have a higher mutation load. And if the tumor is, maybe earlier in its growth, you'll have a lower concentration of the mutation. So with the wild type sensing CRISPR RNA, we of course get detection for all those conditions because there's a large amount of the wild type always present. But for the mutation, you can see we see a dose-dependent increase in the signal. And so this allows us to calibrate sort of signature for the tumor where we can actually detect a single mutation, which I think is really powerful because these CRISPR enzymes are so sensitive to mutations, which is what makes them so specific for genome editing. But we're basically leveraging that specificity for nucleic acid detection now, which is really, I think, powerful. So that was our first sort of iteration of Sherlock. And a lot of our work in the past year and a half has been how can we make Sherlock even better, even more portable. And so Jonathan is actually going to go over that work now. So I'd like to bring him on to the presentation. So.
Great. Thanks, Omar, for that great outline of the Sherlock technology. And once we had completed this initial Sherlock development, we asked ourselves, how can we make the Sherlock technology more useful? So we focused on 3 areas: multiplexing, being able to actually read out multiple different analytes in a single reaction; quantitation, being able to measure not just presence or absence, but the actual amount of a sample in a reaction; and a visual readout. How can we actually create a totally instrument-free platform that doesn't rely on any sort of reader at all? So first, how can we detect multiple targets simultaneously? And this approach really comes from the idea of this diversity that Omar discussed, that there are many different types of enzymes out there that can do this cleavage. So we wanted to find orthogonal Cas13 enzymes to explore new families, as well as use Cas12 enzymes that can cut DNA instead of RNA, which allows us to read out both DNA and RNA in our reporter readout. So the Cas13 family is a very diverse set of enzymes. You can see here that not only are there Cas13As, which we initially discovered, but we also subsequently discovered Cas13Cs and Cas13Bs. And these enzymes differ in many different ways. For example, the Cas13Bs do not associate with the Cas1 and Cas2 accessory enzymes for adaptation. But one thing that we wanted to profile is how these enzymes cut in a different way. So instead of assuming that all these enzymes would just cut RNA, we wanted to profile which bases they would cut. So you can imagine that certain enzymes would cut A and not U, or G and not C. So what we did is we created reporters that were homopolymers, each of A's, C's, U's, and G's. And then we took a large diversity of both Cas13A as well as Cas13B enzymes and assayed them against these 4 reporters. And here you can see that some enzymes actually do have pretty strong preferences in certain directions. For example, the Cas13A that we were using from Lepichikawade, LWA Cas13A over there on the left, has a very strong preference for poly-U's. However, if you look at other enzymes like PSM Cas13B, which is 6 from the left, you can see that it does not cut U's, but instead prefers to cut A's. So if you put these 2 enzymes together in the same reaction and gave these A and U fluorophore reporters different colors, you'd be able to read these out orthogonally. We could actually extend this concept even further. So instead of relying on single bases, we can actually look at dinucleotide preferences. So here we have 2 nucleotides, one after another, with DNA in the other areas. So the enzymes can only see those 2 nucleotides. And here you can see that this can even further separate enzymes. So if you look at LWA Cas13A and CCA Cas13B, initially, when you look at the homopolymers, they both cut U's. But you can actually see that LWA Cas13A will cut AU, while CCA Cas13B will cut UC instead. And so this allows us to actually have 4 different enzymes you can see that cut each a different dinucleotide. Lastly, we can actually supplement the RNA cutting with DNA cutting as well. So Omar mentioned CPF1, which is later renamed to Cas12a, which we discovered as part of this expansion to new class 2 systems. And it was subsequently demonstrated that Cas12a also had the collateral cleavage, except of course because the DNA cutting enzyme on DNA rather than RNA. There is one caveat, which is that when just using the enzyme alone, it's actually much less sensitive than the RNA cleaving enzymes. So here you can see LWA Cas13a and PSM Cas13b, and they can find, you can actually detect signal down to about 1 million molecules for about each of them. But if you look at Cas12a, it's actually much less sensitive. Now, the important thing is if we actually do pre-amplification beforehand, so the entire Sherlock workflow, then all of these enzymes are capable of detecting down to the single molecule level, which means that they can actually all be used for detection. So what we wanted to do is actually create a detection system that could read out 4 things orthogonally. So we took 3 RNA cutting enzymes, PSM Cas13b, LWA Cas13a, CCA Cas13b, and AsCas12a. And we gave them each a different fluorophore and RNA or DNA reporter. And then we each had them target a different molecule, Zika RNA, Dengue RNA, or the synthetic ssRNA1 or dsDNA1. And we looked for detection in each separate channel. So in the absence of any sort of target in the solution, you can see that we have no detection of anything at all. Then if we add in each target individually into a reaction, you can see that we can detect each of them in their own channel and no crosstalk to the other channels. Lastly, you can actually see that for every possible combination of the 16 combinations that have either presence or absence of the 4 targets, we can actually detect that without any sort of crosstalk. So this allows us to have a completely orthogonal way to look up to 4 different colors. And so 4 different potential infectious diseases or 4 different variants that we can all assay in the same reaction. So our second question was, how can we actually quantify sample concentration instead of having a absence presence marker? So we actually came back and looked at that RPA, the recombinase polymerase amplification, because we wanted to tune it so the isothermal amplification wouldn't reach a regime where it was not linear anymore. So what we did is we actually did RPA with different amounts of primer to tune the efficiency. And what we found is that when we use different amounts of primers, too much primer or too little primer actually resulted in a non-quantitative regime. However, when we had just the right amount of primer, we could actually detect multiple orders of magnitude. So we could actually distinguish between orders of 10 all the way up to 10,000 molecules using just a very tuned primer concentration, which allows us to have all of these different assays tuned for quantitative as well as binary readout. Lastly, we wanted to actually generate a visual readout, a method that we could use to have an instrument-independent way of finding if a sample was present or absent. So what we turned to was lateral flow strips, akin to a pregnancy-like readout. So those often take a FAM and a biotin molecule and have the biotin attached to a strip, and we can detect the FAM by the presence of an anti-FAM antibody. So what happens is that if we take a FAM and biotin molecule and put a piece of RNA between them, when there's the presence of the target, it'll cut this apart. And so then when we put this on the strip, if there is no target, so this is a negative test where the reporter is attacked, it'll flow up and reach a streptavidin line, and it'll be stuck there, and then it will bind an anti-FAM antibody. So the antigen will trap the antibody at that line. Now, in the case that we actually do have the target present and the reporter is cleaved, a positive test, well then we won't be able to capture all of that antibody, and it'll actually continue to flow up and flow to a second antibody capture line. This allows us to turn any test that we could normally do with fluorescence in the original Sherlock publication into a lateral flow-based test. For example, for our Zika dose titration, you can see that we can actually turn our Zika detection of RNA into a lateral flow by changing the reporter. And here you can see the strong top band down to about 200 molecules. And then for 20 and 10 molecules, it's a little more difficult to see here, but we actually still have quantitatively increased over the zero control for these conditions. So this allows us to basically make a completely portable test that can detect any nucleic acid of interest. So we wanted to apply these new capabilities to samples. So we'll touch on three things today. One is liquid biopsy of circulating DNA in cancer with our lateral flow assay. Another one is direct Zika detection without instrumentation or purification. And the last is an agricultural application. How can we genotype crops in the field in a very rapid fashion? So first, liquid biopsy detection with lateral flow. How can we actually turn SHERLOCK into a portable way of testing for a burden of mutations associated with cancer? So what we did is we actually took liquid biopsy samples and had them DNA extracted by our collaborators at the Brigham and Women's Hospital. And then we could take that cell-free DNA and do a rapid RPA and subsequent detection. And then we can read it out by fluorescence or by lateral flow. So as Omar demonstrated with the initial samples in our first paper, we can be very, very sensitive to single mutation. So here we can sense the EGFR L858R mutation. You can see that our mutant sensing CRISPR RNA is strong in the mutant condition, but is absent of the wild type condition. So this is fluorescence. But if you actually turn this test into a lateral flow test, you can still see that there's a pronounced band in the mutant patient for the mutant CRISPR RNA that's not present for the wild type patient with the mutant CRISPR RNA. And you can see the quantitation next to it. And we can actually apply this to multiple different cancer-associated genotypes. So here is an exon 19 deletion from EGFR as well. So this allows us to have a very portable method for oncology that's minimally invasive and could be done at a minute clinic or in a doctor's office. Our second foray into rapid and direct detection was in collaboration with Pardis Sabeti here at the Broad and Cameron Mirvold and Catherine Frigi, two members of her lab. And it was about detecting Zika in the field. We had the cover of Science with her on this, and that's a picture of the Zika virus in the dirt in fields. So it's a little bit tongue-in-cheek. But the idea was, how can we detect Zika not only in a very rapid fashion, but with minimal even extraction? So first, what Cameron, Catherine, and Pardis did is that they detected, using SHERLOCK, Zika and dengue virus from patient panels, can compare them to the state-of-the-art controls. So here are samples that were deemed positive or negative from the Altona test, which is a RT-qPCR test for Zika. And you can see that they agree very well, both on these Zika samples as well as these dengue samples for serum clinical isolates and culture dengue. So this is very nice, but commonly for nucleic acid detection, you need to still extract those nucleic acids. So what Cameron and Catherine did is that they developed a complementary method to SHERLOCK called Hudson, which uses nuclease inactivation and viral inactivation heating steps. So all it requires is simple heating and a small amount of buffer, no columns or complicated purifications, to extract nucleic acids and inactivate nucleases that would cause the SHERLOCK test not to work. And then you can put that into SHERLOCK. And they demonstrated that this is compatible with many different common biomatrices. So here you can see that we have PBS and serum. And in serum, you can still detect up to about 100 molecules without purification. In saliva, you can detect down to basically a single molecule regime still. And also, you can do the same with urine. So here we can essentially detect the presence of any sort of biomatrix and nucleic acids inside of that. So they wanted to apply this to samples of flaviviruses, Zika and dengue that they had. So what they did is they took these unpurified samples and they took patient serum and healthy serum. And what you can see is that they have substantially increased fluorescence for the patient serum versus the healthy serum, and the patient saliva versus the healthy saliva. And not only was this compatible with fluorescence, but it was also compatible with lateral flow. So here you can see that for the patient serum and patient saliva samples, we can get strong top bands that don't appear in the healthy saliva, healthy serum, or no input bands. They also developed a test for pan-flaviviruses. So using universal RPA primers, you can actually determine the presence of Zika, dengue, West Nile, and yellow fever. So by designing guides against each of these different flaviviruses, you can actually determine orthogonally the presence of each of them without detecting the others. So of course, in combination with our fluorescence, our multiplexing that I discussed earlier, you can imagine a in-sample rapid test for all of these different potential flaviviruses. And you can see that they're totally orthogonal. So these are samples with actually 2 flaviviruses per. You can see that the actual detection matches exactly what the input samples were. Even beyond this, we can detect single base mutations in Zika patient samples. So in the evolution of Zika, the Honduras strain is more divergent from the Dominican Republic and United States strains, and you can actually detect the presence of these different single base mutations that cause them to be defined. So you can actually see that patients with samples from the Dominican Republic versus the USA or Honduras correspond to their actual mutation samples compared to the ancestral seed stock. So we can actually genotype different strains to figure out where they're from. But even beyond that, we can actually do genotyping to figure out important features for pathology. So many people think of Zika as being strongly related with microcephaly, but there's actually the evolution of a single mutation in Zika that contributes to this microcephaly phenotype. So we can actually potentially detect that. So during this study, this mutation was found. And the nice thing about the SHERLOCK platform is because it's so programmable and so robust, we can actually design the samples, the CRISPR RNAs and the RPA primers, and design the entire assay and test it in under a week time. So what Cameron and Catherine did is they actually designed CRISPR RNAs against the target as well as the ancestral stocks. And you can see that the ancestral stocks and target stocks can be distinguished based on the presence of these mutations. And the microcephaly-associated mutation can even be read out in the lateral flow format. So you can see the derivative and seed blocks all have the derivative but not the ancestral, and vice versa. So we can actually find relevant mutations very rapidly and without instrumentation. Lastly, we can apply this to agricultural genotyping, literally in the field, in situations where you may want to rapidly look for pathogens or other plant properties. So what we did is we wanted to look for the presence of Roundup Ready crops. This is the CP4 EPSPS gene, and we wanted to compare that presence to the lectin gene, which should be present in all soybeans. So we took soybean samples and ground them up in a rapid extraction assay, and we found that we could actually detect substantially more of the EPSPS signal in the samples, but only when the GMO was present. So we actually took a rapid cDNA extraction, and we took soybeans, did a rapid RPA, did a 20-minute Cas13 reaction, and applied lateral flow detection. And we were able to actually, within 30 minutes, be able to detect the presence of the Roundup Ready gene in these soybeans compared to the wild type. And this was substantially increased when we quantified it. So here you can see that we've improved on many applications and properties of the SHERLOCK platform. We've increased multiplexing by using multiple different enzymes. We've improved with quantitation by tuning primers. We've used lateral flow detection to actually create a completely visual readout. We've been able to use amplification to detect additional signal using CSM6, which is discussed in our paper. And we can actually do direct detection with crude samples using Hudson. So this in all creates a very portable and rapid pipeline for detecting nucleic acids across multiple different applications. So SHERLOCK, the technology, is still, we think, in its infancy. We're working with many collaborators to continue pushing the platform forward. And if you're at all interested in using the platform, either for academic or commercial use, please reach out to us, because we are working on creating a large portfolio of resources for people to use the tests, which I think can be incredibly useful for the community. So with that, we'd like to thank all the people who are involved with this work, who made it possible. And this is a picture of the Zhang lab at the Broad Institute. And none of this would have been possible without our mentor, Feng Zhang, who created an incredible environment for this and who was instrumental in every step of the process for creating the SHERLOCK technology. But of course, we also want to thank the people who helped us at the bench, Max Kellner and Jeremy Koob and Patrick Usick-Bickler, three master's students, and Jeremy is a research associate who were vital in helping out with the actual development of the SHERLOCK technology, as well as our many collaborators, Jim Collins, who initially helped us with the development of the fundamental SHERLOCK test, Pardeep Sabeti, who you saw helped us with many infectious disease viral applications, Deborah Hung, who has been instrumental in bacterial applications, and Eugene Koonin and Kira Makarova from his lab, who have been very helpful with our exploration of biodiversity throughout this. So with that, I'd like to thank you all for listening.
Well, thank you, gentlemen, for that informative presentation. And I would also like to thank Labyrinths for making today's educational webcast possible. And as a final reminder, your speakers will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.